Hello everyone, I'm Dr. DP Bora. Dear friends, I welcome you all to my YouTube channel, The Microbiology Digest. Today I'm again back with a new topic, which is one of the very important topic to understand, that is structure of bacteria. So whenever there is discussion about the microbial world or the different processes involved in microbiology. So first of all, it is very much essential to understand the different components, or as a whole, the structural details of an organism. So here in this discussion, today's discussion, we will be discussing on the topic which is very important and very much essential to understand microbiology as a whole. It is the structure of bacteria. So first, a brief introduction about bacteria. Already we have discussed in our previous discussions that bacteria are prokaryotic microorganisms. So they lack two true nucleus and all membrane bound organelles, because they don't have a true nucleus and membrane bound organelles. Cell wall of bacteria, it shows very much simple, or it is said to be very much simple in structure, but it carries out so much different functions. So it is said as it possesses the structural simplicity as well as functional diversity. So bacterial cells are smaller and simpler in structure than eukaryotic cell, that we all know, we have already discussed in our previous discussions. They don't have clear membrane bound nucleus. So it is well understood, because already we have in our previous points we have discussed that there is no true membrane bound nucleus or organelles. So now, coming to the bacterial structures, bacterial cell, if we consider one bacterial cell, so we can divide different structures present in a bacteria into three different parts. First, it is external structures, which includes the appendages and the coverings. So under this, we have the glycocalyx, which is sometimes it is also called as capsule. We have the flagella, we have the Fimbriae, then the Pilli, or if it is single, we can say it as a pilus. So next part is the cell envelope. So under cell envelope, we have two different cell envelopes. One is cell wall, second one is cell membrane, or in some cases it is also otherwise called as cytoplasmic membrane. Next part is the internal structures. So internal structures, it includes the cytoplasm, nucleoli, plasmid, ribosomes, mesosomes, and the storage granules. These are the different components which are present in a bacterium, and for easy understanding, we have divided these components into three different parts. First part is the external components or external structures, including the appendages as well as the coverings, then cell envelope and the internal structures. So in our subsequent discussions, we will be discussing one by one structure in greater detail. Now here, this is the typical diagrammatic illustration of a bacterial cell. So here you can see So if we as we have divided into three different parts. So first is the capsule. So this is the outer layer. In some bacteria, this is not an universal component of bacteria. In some organisms, it is present. So this is the capsule, or it is also sometimes it is called as glycocalyx. Next component, which comes is the flagella. Flagella, this is the flagella. You can see this is the flagella. Then we have the Pilli or Fimbria. These small appendages, these are all called as Fimbriae, and there are some different, structurally different and small in number. They are called pilas or Pilli. Then coming to the next component, it is the cell wall. So cell wall, it is the external covering in most of the bacteria. Then next covering is the cytoplasmic membrane, or it is written as plasma membrane. It is same. It can be said as cytoplasmic membrane or plasma membrane or cell membrane. So these two are the membranous structures which are present in bacteria. Then the internal structures which are present in bacteria, they are, first one is the nucleoid, because this is not a true nucleus, just which is in contrast to that one present in eukaryotic cells. It is not a well-defined structure. So instead of nucleus, it is called as nucleoid in case of bacteria. Then we have the uh, plasmid, we have the ribosomes, then we have the inclusions, then we have the mesosomes. So these are the internal structures which are present in bacteria. So for now, one by one, we will discuss in detail. First external structure is the glycocalyx. So glycocalyx, it is the it is found, as I said, it is not an universal structure in bacteria. It present in only some bacteria, and it is a slimy or gummy substance. When it is present, that becomes the outer covering of that organism or bacteria. When it is present, it becomes the outer covering. And in most cases, the glycocalyx, it is composed of polysaccharides. Now, there are some other terms which are also uh, included under this glycocalyx structure. Sometimes it is called as the capsule. So when we say it is a capsule, when this glycocalyx layer, it is a thick and well-defined and thick glycocalyx structure, which when it is present in an organism or bacteria, then it is said as capsule. And in most cases, that capsules, it is composed of the polysaccharides. But there are some organisms, for example, in case of Bacillus, so it is composed of the polypeptide. So Bacillus organisms, exceptionally, instead of uh, capsule is made up of polysaccharide. In case of Bacillus organisms, the capsule, it is made up of polypeptides. Polypeptide is the poly-D-glutamic acid. Then when this glycocalyx layer is a thin one, and it is just exists as a polymeric material, and it is a loose meshwork of fibrils around surrounding the cell, it is called the slime layer. So in some organisms, the glycocalyx layer, it may exist as a slime layer, and in some organisms it may exist as a capsule. So what are the functions of this structure? It protects the cell from drying. It is very, very important in those organisms where it is present. As I said, this is not an universal structure. So when it is present, it protects the cell from drying. Then it prevents the attachment of phages. Phages, we all know these are viruses which can destroy the bacterium. So these capsules or glycocalyx or slime layer, when it is present in whatever form it may be, so it will prevent the attachment of phages. But on the other hand, again, same structure, it helps the bacterium to attach to some surface, because in the infection process to start, so it is very much essential for the bacterium to first attach to the surface. So this is that this layer, glycocalyx layer or the capsule layer or the slime layer, it helps in attachment of the bacterium to a particular surface. Next, it gives protection to the antibacterial agents in organisms when it is present. For those organisms when it present, so it gives a protective covering so that antibacterial agents, they cannot destroy the organism. So it acts as a overall protective structure. So again, it gives resistance to the phagocytosis. So when it enters to any host system, so immunological reaction starts, and phagocytosis, through phagocytosis, our body tries to eliminate these external agents. So this capsule, it protects the organism from the phagocytosis. Then last function of this structure is the it provides immunological specificity to the bacterial cell, because these capsules or glycocalyx, they may contain some uh, regions which can give immunological specificity to the organism. Okay, then coming to the next structure, it is the flagella. Flagella, as I said earlier, this is the locomotory organelle of bacterium. With the help of flagella, bacteria attains the capacity to move from one place to another. Means, in other words, bacteria becomes motile with the help of flagella. Okay, so these are slender rigid structure about 20 nanometer in diameter and 20 nanometer 20 micrometer long. It is a very long structure. It becomes many times than the bacterial organism. Okay, so this flagella, it is composed of a specific protein. The name of the protein is flagellin. So flagellin is the structural unit of the bacterial flagella. So bacterial flagella, it is composed of three components. It is a, as I said, it is a very long structure. It is having three different components. One is basal body, then second is hook, and the filament. So in the diagrammatic illustration, you can see this is the filament structure, this is the hook, L-shaped hook, and this is the basal body. So basal body, as depicted in the diagram, it is having four different rings. These rings are named as M ring, S ring, P ring, and L ring. Okay, so specific names why this has been named. It is written M ring, it stands for the membrane ring, because this part, basal body, it is actually, it is the structure which is present in the cytoplasmic membrane. So the hook of the flagella, it is it acts as a universal joint between the filament and the vessel body. Means the vessel body, it remains in the inside the cytoplasm, cytoplasmic membrane. Then filament is a different structure which is very much long, and these two structures are connected by the hook. And as I said, it is the longest and the most obvious portion of the flagella is the filament. Okay, so filament, it is the last component of the flagellum after basal body, then hook, then then it is the filament part, which is very long. Now, depending on the distribution of the flagellum in bacteria, so the pattern in which flagella are distributed. So on that basis, we can have different types of arrangement in case of bacteria. First is the monotrichous. Monotrichous means bacteria having only one flagellum. Okay, it may be the flagellum or flagella may present in one end, or sometimes it is said as polar flagella because it is having only one flagella at one end or one pole. It is said as polar flagella. Then we have the lophotrichous. So lophotrichous means, lopho means a tuft or a bundle. So when a bundle or tuft of flagella is present in one end, okay, or in both the ends, it is called as that those bacteria having that type of flagella are called as lophotrichous. Okay. Then third is the peritrichous, when flagellas are distributed throughout the surface of the bacterium. It is called as peritrichous. And amphitrichous is the arrangement of flagella when flagella are distributed one flagella at each pole. Means both the ends will have one flagella, and this type of organisms are called as amphitrichous. Then one more term is there, I have not mentioned here, that is the atrichous. Atrichous. Atrichous is a term given to those organisms when there is no flagella present in a bacteria. That condition is called as atrichous. Then when, as I said, flagella is the locomotory organelle of the bacterium. So whenever there is presence of flagella, the bacteria will be motile, because this is the principal function of flagellum is to help in the motility or movement of the organism. And one surprisingly, when the flagella of bacteria grows, it does not grow in the base, it grows in the tip. This is very important point to understand. Whenever there is growth of flagella, always it grow at the tip, not at the base. Okay. So in the diagram you can see this is the monotrichous flagella bacteria having only one flagella. This is the lophotrichous, a tuft of flagella. Then this is the peritrichous, flagella distributed throughout the surface area. Then this is amphitrichous. Okay. Next is the Fimbriae, or it is also otherwise called as Pilli. So these are fine, straight, and hair-like appendages, and these structures are visible only under electron microscope. So typically they are shorter than the flagella and more numerous. So whenever there will be flagella, so specific numbers will be there. But Fimbria or Pilli, they are more in numbers than flagella. So like flagella, here also these structures are composed of the protein pilin, and they are attached to the cell wall of many bacteria. In most cases, so these pili or Fimbria, they are attached to the cell wall. So in the in organisms or in bacteria, we may encounter two different types of pili. Type 1 is called as longer pili, or second one is shorter pili. So those who are longer pili, they are said as sex pilus or sex pili. They mostly occur in or they mostly take part in the conjugation process when there is uh, transfer of genetic material from one organism or one bacterium to another. So the function of sex pili is to take part in the conjugation process. What happens in conjugation? There is transfer of the genetic material from one organism to another. Now, what is the functions of pili? Most important function of pili is they helps in attachment or adhesion of bacteria. Okay, so whatever uh, Fimbriae are mostly having this function. The attachment structures, they are the Fimbriae, and the pilus, they take part in the conjugation as I have discussed earlier. Then next important, very important structure in case of bacteria is the cell wall. So it has a rigid, solid covering that provides shape and structural support to the cell. So bacterial cell, it receives the structural support from the cell wall. It lies between the cytoplasmic membrane and the capsule if it is present. If the bacteria they are capsulated or capsule is present, then the cell wall lies just next to the capsule internally. So periplasmic space is the space between the cell wall and the cytoplasmic membrane. Okay. Then the cell wall, it is a dynamic structure. The thickness of this structure, it varies from bacterium to bacterium, and the cell wall, as I said, it is one of the very important structure in bacteria, and it may contribute from 10 to 40% of the dry weight. You can imagine overall weight of the bacteria, dry weight of the bacteria, 10 to 40% is given by the cell wall. Okay. Now, what are the functions of the cell wall? It, as I said, it imparts the shape and rigidity. It supports, it gives support to the cytoplasmic membrane because cytoplasmic membrane lies next to the cell wall. Internally, it lies next to the cell wall. And this cell wall actually, it protects the bacterium from mechanical damage. Okay. So mechanical damage, it gives the protection from mechanical damage. And the osmotic lysis, in case of bacterium, there is always a problem of osmotic lysis. So the cell wall is the structure which protects the bacterium from osmotic lysis. And in some organisms, the cell wall, it contributes to the pathogenicity of the organism or bacteria. So these are the four important or functions carried out by the cell wall. Now, there are differences between the cell wall composition. So, cell wall composition is very essential to understand. So, basically, the main component of bacterial cell wall is the peptidoglycan. So sometimes it is also called as murin. Peptidoglycan or murin. It is a mass-like polymer and it is very, very unique to the prokaryotic cells. So peptidoglycan is present only in prokaryotic cells. Now it consists of, as I said, it is a polymer. It consists of uh, glycan chains mostly, and two alternating units are present. One is called as N-acetyl glucosamine. In short, it is written as NAG or NAG. And second one is the N-acetyl muramic acid, or it is in short it is written as NAM. Means peptidoglycan, it is a polymer of two glycans. One is N-acetyl glucosamine and other one is N-acetyl muramic acid. Means it is a polymer consisting of mainly NAM and NAG. Okay. So these two subunits, they they exist in repeating units, means alternating units of each NAM and NAG, and these alternating units are linked, cross-linked by some tetrapeptide as well as pentapeptide cross bridges to give a mesh-like structure to the cell wall. So in the diagram you can see, so this is the N-acetyl muramic acid, the round structure, and this is the dark structure, it is the N-acetyl glucosamine. So these two structures, the glycans, they exist in alternating subunits. This is uh, N-acetyl muramic acid or NAM. This is NAG, again NAM, NAG, then NAM, NAG. It continues alternatively. First NAM will come, then NAG, then NAM, NAG. It will continue in a chain system in an alternating manner. So these two structures, these two glycans, NAM and NAG, they are cross-linked to each other by tetrapeptide side chains. Each NAM and NAM, they have a tetrapeptide side chain and they are cross-linked to the other by pentapeptide bridges. So this is the NAM, which is having a side chain of four different peptides, tetrapeptide side chains, and this is another NAM or N-acetyl muramic acid having our four different peptide side chains. So these two side chains are cross-linked to each other by a pentapeptide bridge. This is a pentapeptide bridge. So this structure, it gives a unique, very mesh-like structure which is present only in prokaryotic to form the bacterial cell wall. So what is the principal structure? So it is the peptidoglycan, which is composed of alternating subunits of N-acetyl muramic acid and N-acetyl glucosamine. These alternating subunits are cross-linked by tetrapeptide side chains and pentapeptide bridges. Okay. So in all bacterial species, the basic structure or backbone of the cell wall is same, but there may be difference in the side chains as well as cross bridges. Okay. So these differences in the structure and chemical compositions so may lead to variation in their pathogenicity in case of bacteria and other important characteristics of the bacteria. So now based on the cell wall structure, so as we all we have discussed in our staining discussions, so there are two different distinct groups have been identified. One group is called as gram-positive organism, one group is called as gram-negative organisms. So this distinction between gram-positive and gram-negative, it is based on the cell wall structure of the organism. So in gram positive bacteria, there's a thick multi-layered peptidoglycan layer. The peptidoglycan layer, which is present in case of gram positive organism, it is very thick, and the entire structure is very rigid, and it gives additional strength to the bacteria. Okay. So peptidoglycan, in case of gram positive bacteria, it may account up to 50% of the dye weight. So 50% of the dye weight of the bacterium, it is provided by the peptidoglycan alone in case of gram positive bacteria. So there are some other structures which give extra rigidity to the bacterial gram positive bacterial cell wall. It is the teichoic acid. Then again, in the membrane, teichoic acids are present in case of gram positive, which is not present in gram negative. It gives additional strength to the gram positive cell wall. So when we will see the structural details diagrammatically, so we will be very much able to differentiate between the gram positive cell wall as well as gram negative cell wall. Next is the gram negative cell wall. So in contrast to gram positive cell wall, gram negative organisms, they have an additional covering. That covering is called as outer membrane, which is mostly composed of lipoproteins or lipopolysaccharides. And here, in case of gram negative organisms, the peptidoglycan layer, it is very thin, because it is having a very high content of lipids. It is ranging from 11 to 22% of the dry weight of the organism. So the peptidoglycan layer is very, very thin in case of gram negative organism. Okay. And again, another structure which is very rich in lipid, that is the outer membrane, that is present in case of gram negative bacteria. So gram negative bacteria, as I said, the outer membrane, it is mostly composed of the lipopolysaccharides. So it is having three different components: the LPS, which is the principal structure of the outer membrane, and also a component of the gram negative cell wall. So it is having three different components. So one is core polysaccharide, then second one is the lipidae, and third one is the polysaccharide O antigen. So these three components, they consist or constitute the LPS moiety of the gram negative cell wall. And as the outer membrane is an additional structure, and it is mostly composed of the lipids, and because of this, gram negative organisms, they are very rich in lipid content. So lipid content in case of gram negative organism is, it may range from 11 to 22% of the bacterial dry weight. Okay, bacterial dry weight, 11 to 22% of the dry weight is from the outer membrane or from the lipids only. Okay, so this is the diagrammatic illustration. As you can see, this is the gram positive cell wall. Here peptidoglycan layer is very thick, and this is the gram negative cell wall. Here peptidoglycan layer is a very thin layer, you can see in the diagram. Okay, so because of this, it is having a very high content. Gram negative cell wall, they have a very high content of lipids. So it ranges from 11 to 22%. Now it is very much essential, or it is very much important to understand the structural differences between gram positive cell wall and gram negative cell wall. So here I have compiled some of the points. So first is the characters, then gram positive cell wall, then gram negative cell wall. So peptidoglycan layer, as we have already discussed, it is very thick in case of gram positive. It is very thin in case of gram negative cell wall. So lipid contents, it is almost nil in case of gram positive. So here it is very high. Then lipopolysaccharide, mostly it is absent, but it is present as endotoxin. So lipopolysaccharide or LPS component of gram-negative bacteria, it is the endotoxin part of gram-negative bacteria. Okay. So what is endotoxin and exotoxin? We will be discussing in our subsequent discussions. Then teichoic acid moiety, this is present only in gram-positive organism. That is absent in gram-negative. Then there are some other differences, and one important is the periplasmic space. It is very thin, or sometimes very less, uh, or sometimes it may be absent in case of gram positive, but periplasmic space is always present in case of gram negative organism. Okay, so this is what in so we have some more different components of bacterium, so we'll be discussing in our subsequent part, because if we go on discussing, the video will be a bit uh, longer. So we'll stop here, because up to cell wall we have discussed today. Then after next to this is the cell membrane or cytoplasmic membrane. So we'll be discussing in those structures in part 2 of this bacterial structure discussions. So I hope this information will be helpful for you all. And if you feel that these informations are helpful, please subscribe to my channel, the Microbiology Digest, and I'll be more than happy if I can receive some comments from you, all my friends, regarding the contents which are included here, or if I missed any portion, please let me know. I'll try to include in my subsequent uh, videos or lectures. So till then, please subscribe to my channel and share my channel with your friends, so that uh, I can get more and more comments from my dear friends, and it will be helpful for me as well to prepare more and more, uh, or to gather more and more informations on the different topics related to microbiology. So with this, so we'll stop here today. Thank you very much, and